Okay, everyone, thank you very much to tune in into this special webinar recorded session that we organized from the NTU Research Center for Epidemic Prevention Science, NTU RCEPS, together with NTU and NUS to bring you this webinar, AI-Driven Drug Re-positioning. Especially for the COVID-19 drug discovery. So today we have two distinguished speakers that I'm so happy that I can put them both together. So the first speaker that we will have is Professor Xuefeng Ran from National Taiwan University. So she's a distinguished professor from Department of Life Science, Graduate Institute of Biomedical Electronics and Bioinformatics. She's also the key director of Taiwan AI Labs she has worked her career in the field of systems biology, and recently she has, in collaboration with Taiwan AI Lab, they have pushed forward the research to create a database driven by AI to discover drugs that could be repositioned for COVID-19. And the second speaker for today is Professor Dean Ho. So Dean, if you think of anyone to be the face of AI for Singapore, that would be Dean. So Dean is a provost chair, professor, director of the N1 Institute for Health, and director of the Institute of Digital Medicine. Wisdom in NUS. He's also the head of Department of Biomedical Engineering. So Dean has an illustrious career as a young faculty. No, when I say young, it's physically young, but he has a family connection with Taiwan. We were just chatting that both his parents graduated from NTU. So I think today this webinar is very special, even though it is the NTU NUS. So we can sort of say that it has NTU blood in it. So without further ado, so let's start today's session. So this is me. I am Ruby Huang, and I'm a professor at School of Medicine in College of Medicine in NTU. My role here is as the lead PI of the international collaboration of NTU RCEPS. So today, our purpose is really to bring together the collaborative, the international collaboration, particularly during the pandemic of COVID-19. And then we want to keep this international collaboration going forward, even after we survive and recuperate from this pandemic, that we can forge a stronger international collaboration. So without further ado, I would invite Professor Ran for the first talk, which is the drug repositioning for COVID-19.
Okay, thank Professor Huang's invitation and introduction. Today, I want to present our recent work on drug repositioning for COVID-19. As you know, AI can be used effectively in different parts of drug discovery, including drug repositioning, drug design, and drug screening. So as an alternative or complementary approach to high throughput screening assays with high cost and low hit rate, virtual screening is an efficient computational method to identify drug candidates from largest compound databases. So today I want to present my work on drug repositioning for COVID-19. As you know, the confirmed cases of COVID-19 disease is close to 100 million. So it is important to find out drugs. From the search in PubMed using COVID-19 as the keyword, we found more than close to 100,000 literature has been reported. And there are more than 4,000 clinical trials ongoing. So traditional drug discovery is through serendipity finding. So, for example, the cattle ate sweet clover and got a sweet clover disease, which is bleeding to death. So the doctor isolate the compounds offering from the sweet clover, and finally found the compounds have the anticoagulant effect. Other famous serendipity findings including penicillin. So drug repositioning is a rapid and cost-effective way to discover new therapy for cure the emerging disease such COVID-19. So drug discovery also can help to discover new indications for existing drugs. So there are two approaches for drug repositioning. One is from a known compound to new target. The other one is from a known target to new indication. So in 2003, I joined the national team led by Professor Wang Qihui. At that time, we know some compounds, some drugs have effect on for anti-SARS. So we use the data mining for similar structural compounds. Then finally, we found more potent drugs, as shown the right side. So in the beginning of last year, I proposed the strategy to our funding agency, MOST. At that time, I proposed how to move this. So I proposed virtual screening and big data analysis. So we can perform docking simulation to find potential drugs. So the docking simulation can against compound database such as Taiwan NHI drugs, FDA-approved drugs. So using big data analysis, we can also find antiviral drugs. So then we validate our prediction using enzyme assays, cell-based assays, and we want to perform animal model. After we got some drugs, we can use drug-drug similarity. As I showed you in the previous slide, we can find novel drug. So to how to select the molecular targets of SARS-CoV-2. From the slide, you can see the spike protein on the SARS-CoV-2 surface can bind to the ACE2 on the host cell surface. Then this protein TMPRSS2 cavitate the interaction. Then the virus enter the host cell. After the virus enter the host cell, some proteins are important for viral production. For example, the PL protease, M protease, DRP, and N protein. So our strategy like this, if we know the protein structure, we directly perform docking simulation against this drug database. If we don't know the protein structure, we first perform the sequence alignment, homology modeling, and then perform docking simulation. So how to select the drugs? In this database, we try to build a drug database against SARS-CoV-2. So we select these 7 drug targets, 5 from the viral proteins, including spike protein, DRP, 3CL protease, PL protease, and N protein. And 2 from the human proteins, ACE2 and TMPRSS2. So our database provides the information, including the docking structure, ligand information, and also the experimental data. So first, we use open barbell to convert the 3D structure of the compounds, then perform the auto-dock VNAT for docking simulation. Then finally, we provide the heat map visualization and the candidate drug list. So to compare our strategy, we found our compounds and the determined 3D drug protein structure, they have the similar binding pocket. So our database also provide user to download the data. For example, they can download the proteins, ligands, and ligand with all process data. We not only provide data set, but also provide the open scripts and source code on GitHub. So if you have interest in proteins or drugs, you can perform docking simulation with similar parameters using our source code. So last weekend, the Discovery Channel have reported our SARS-CoV-2 database here. So the second part, I want to show you how to use big data analysis to discover interferon beta-induced host signatures and reproduction drugs for COVID-19. If the host have a high innate immune response, it leads low viral production, lower symptom, and low inflammation, and become a good prognosis. If the host have low innate immune response, it leads high viral production, hyperinflammation, high severity, then become death. So the coronavirus stimulate the production of pro-inflammatory cytokines and type 1 and 3 interferons. So the secrete interferon bind to the receptor and induce hundreds of antiviral effectors or interferon stimulate genes to achieve a cell intrinsic state of viral resistance. This slide shows low viral load cause early interferon response and rapid viral clearance. If the high viral load will cause a delayed interferon response, viral persistence, inflammation, and finally become severe disease. So we propose a hypothesis like this. Compounds enhance the host response to interferon may be effective against SARS-CoV-2 infection. And we use a gene expression-based approach to make such a prediction. So these figures show our schema. First, we identify the interferon stimulate gene signatures, then use our developed methods to predict the potential drugs. Then finally, we perform a cell-based assay to validate our prediction. We collect the data from the GEO. Then we use our principal component analysis of the host RNA-seq data. As the results show here, these three data sets have the conditions related to interferon beta treatment. And we also measure the correlation of host transcriptional response. And the result, similar with the previous slide, these three data sets are correlated response of interferon beta treatment. So we compare these three data sets and identify more than 1,000 genes with interferon beta gene signatures. Then we use these 1,000 genes to perform functional and pathway enrichments. So the bigger figure B shows the top 10 pathway. For example, the interferon signaling, interferon alpha beta signaling. So next, we use our developed system level method for drug discovery. Here show the perturbation means the chemicals and cross many genes and cross various cell lines. So for example, this compound activate the genes and the other two compounds inhibit the gene. We try to give the score. So I will show you how to give the therapeutical score in the next slide. So we use the connectivity map data, which is a genome scale library of cellular signatures that catalog transcriptional response to chemical, genetic, and disease perturbation. So in this study, we use the second generation of CMAP called as links. So links data contain over 1 billion data points. So there are many perturbation types, for example, the small molecule or antibodies, and contain 5 cell types, including more than 1,000 cell lines, and 5 signature types, for example, the transcriptomics data or proteomics data. So in this study, we use the transcriptomics data to inform what kind of the chemical can have the potential against SARS-CoV-2. So this slide shows how to give the expression-based therapeutic score. For example, G means gene, plus means upregulate, this gene upregulate. P means perturbation, for example, chemicals or compounds. And minus means inhibit. D means differential express gene, plus means upregulate. So if the compound inhibits the gene expression, we call this reverse hit. If the compound activates the gene expression, for example, this gene upregulate, then the compound enhance the gene expression called as forward hit. So in this study, we try to identify the compounds which can stimulate interferon gene signatures. So we give the score like this: summarize the forward hit, subtract summarize the reverse hit. So the data, the results show here. Figure A show predict compounds which can enhance the interferon beta signature. Then B show the compounds are associated with interferon beta 1 regulatory recurrence. So we try to overlap these two data, then found 4 compounds have the potential against SARS-CoV-2. The next, we try to construct the drug target network. For example, this drug inhibit the gene expression and enhance this pink gene expression. And also, we found these 4 candidate compounds induce a similar host response of interferon beta, so provide a molecular mechanism for their potential efficiency against COVID-19. So B show we also perform the functional and pathway enrichment and found the top 1 pathway is top 1 interferon production. The top 2 is immune response regulatory signaling pathway. So we use a plaque reduction assay to validate our results. So first, we infect the cell, the viral E6 cells, with the SARS-CoV-2 virus. After 1 hour, we treat the drugs for incubation for 5 days. Finally, we quantify the results. So our results show here, these compounds have the best one in IC50 around 16 nanomole. So I want to summarize my talk. We developed the COVID-2 drug database to predict the potential drugs with validation information support. And we identify the interferon beta induced host signatures and repositioning drugs for COVID-19. So in our study and validation results, we found 3 drugs with the best inhibitory concentration below 20 nanomole. So finally, I want to thank my collaborator, Professor Chen Qianyu, Huang Xuancheng, and Zhang Shuyuan. And also, I want to thank my postdoctor, Huang Zhenzhong, Xie Qiaohui, and my PhD student, Huang Chenghao, and the team members in AI Labs and the funding agency. Thank you.
Thank you, Professor Yuan. Professor Ran, sorry, that was, I think you gave us a very good example of how the novel discovery can actually still be made through many readily available public resources, and then to have a very quick response to public to meet with the public emergency like the COVID pandemic. Yeah, so we will keep the discussion to the last. So we will introduce Dean's talk first, and then we will leave several, probably 5 to 10 minutes for kind of like a cross dialogue among us. Okay.
Okay.
So our next speaker is Dean Ho, and he's going to tell us how, from his experience in NUS, how they utilize also the power of digital medicine to optimize drug repositioning. Dean, up to you.
Great. Thank you so much for the kind invitation. It's great to see everybody. I'm going to share a kind of a spectrum of ideas that we've been working on, and it was a wonderful talk that we just had. And what I'm going to talk about will kind of lead into how we can develop repositioning or novel candidates. But I kind of wanted to take everybody through some of the work that we do that spans everything from drug selection to how we dose these drugs clinically. I'm going to start off with a very brief introduction of what we do at the two institutes, and then talk about some of the challenges in development that we've been encountering as a field, and then give a couple of use cases across not only COVID, but some other applications as well. I wanted to start off by acknowledging the individuals that make this work happen. So N1 and Wisdom are both institutes that are clinical stage. So 99% of what we do are clinical trials with whether it's AI, digital medicine, but also completely reimagined clinical trial design. And this happens with a team of amazing clinicians, electrical engineers, cognitive psychologists, physiologists, and beyond. And I wanted to point out Mihul, who is an electrical engineer, but works in areas like incentives building to ensure that whatever technologies we use, there's incentive for providers and patients to continuously use the technologies that we develop. And for technologies to be truly implementable and sustainable in healthcare workflows, we bring in people like UC from NUS Business to handle operations and analytics research, as well as healthcare economics. And I'll talk a little bit later about how our infectious disease work has involved him and other team members as well. The core ethos of N1 and Wisdom are that technology alone cannot transform healthcare. And that's important because it really does take a spectrum of stakeholders, from tech development to biology to clinicians to healthcare econ, behavioral sciences, and beyond, to really bring technologies into redefining how medicine is practiced. So I'm going to start off by talking about some of the challenges with drug development in general. Now, if we look at novel therapeutics, especially for oncology, the cost of bringing new drugs forward is, as we know, very expensive. And reasons include that the fact that they take too long to develop, success rates are low. And in particular, when we think about new drugs or repurposed drugs, they often end up in combinations. And when developing combinations, it's really difficult to really know which drugs should be put together at which respective dose. The predominant notion in drug development is that if a compound is promising, whether it's repurposed or whether it's novel, that even if the dosing is incorrect, in particular for combinations, even if the dosing is incorrect, if the drugs are good, you'll get some efficacy. But as we've shown clinically, that's not the case, where even good drugs given at the wrong dose can actually have no efficacy. And what this actually means is there are patients who could respond to this therapy. Just by changing the dose, you can actually reacquire efficacy. And so we are in fact missing patients that are responders because of standard fixed dose regimens. What this shows us is that when we develop combinations, we have to simultaneously account for which drugs to give and which dose for each drug, because that can have a profound impact on which drugs we pick in the first place. And I'll show you examples of that in a little bit. As we know, drug development, when done suboptimally, leads to substantial costs for patients. Zolgensma, 2.1 million, Luxturna, 850,000. Makena used to be $20, then became $1,500, culminating in tens of thousands of dollars in efforts to prevent preterm birth. Unfortunately, even though drugs cost a lot to develop, they don't always work very well. Not long ago, an FDA advisory panel advised to take Makena off the market. Many patients have spent a lot of money to take this drug only to have it not work potentially. And so I think that when it comes to developing better therapies, we have to do it optimally, and then in so doing, we'll be able to bring more value to patients. So to do that, I think we have to completely rethink how drug development is done. As the wonderful talk before showed, there's a lot of cool work being done on identifying repositioning candidates. And in the last year, we've seen a lot of cool work from large-scale compound repurposing to developing novel assays for repurposing. But again, the repurposing stage of development represents what we know as discovery, right? Target ID, lead generation optimization, as well as identifying repositioning candidates. But it's really important to know that drug discovery and drug development are very different parts of the roadmap. Both necessary, both should be seamlessly integrated, and there's the component of administration, which is after you have the drugs you want to use, how do you dose them? And as we've seen, dynamic dosing versus fixed dosing can have a profound impact on sustaining positive patient outcomes during treatment. All right, again, when people think about AI and drugs, they largely think about discovery, but there's a lot of work that's got to be done to make sure that the drug is shepherded forward properly. Development includes many things. It includes combo design, it includes matching patients to trials, and this is done to ensure that when you have a promising drug, you co-deliver with the right candidate compounds at right respective doses. To carry on what I just said, I was talking to a friend at Eli Lilly a couple of years ago, and I asked him a question. I said, in big pharma, to what extent does dosing matter when you're doing combination therapy? And he said, of course it's important. You want to get the right amount of drug to the right disease site, and you want to minimize toxicity. But then I said, that's not what I'm asking. I said, I'm asking, to what extent does knowing how to dose combinations help you pick which drugs you put in the combination in the first place? And to that question, the drug developer, he knows the work that we do. He said, Dean, I understand what you're asking. And he was like, we don't consider that at all, right? Remember, picking the right drugs and picking the right dose has to be done at the same time, because solely using mechanism-based drug selection, as we know from decades of drug development alone, cannot solve the issue of the probability of success, right? And so when optimization, this is actually much more of an optimization thing than it is just pure AI for our work, right? Picking the right drugs and the right dose has to be done simultaneously. Because if you take the traditional approach of pick the drugs, then find the dose, that is implicitly suboptimal, right? Because optimization is really about space, drug and dose parameter spaces, and reconciling both at the same time. So I'm going to give you an example of what I mean by this. We often get asked questions. People come and say, hey, look, with AI, and I give you a bunch of data, can you help me find which drug combinations would be best for a certain disease? So people say, look, is drug 1 and 2 the best possible combination for a specific disease? Somebody else can come and say, hey, look, is drug 1, 2, and 3 a better combination? Someone else can even ask me, is drugs 1, 2, 3, and 4 even better than drugs 1, 2, and 3 put together? And people can come and say, I've got millions of data points of different patients getting these regimens. Can you tell me, is the doublet, the triplet, or the quadruplet regimen better for patients? Can I predict? Here's the problem. No matter how many millions of data points you get, these data points are often based off of fixed dose, right? And so no matter how many billions or millions of data points we get, it's very unidimensional, right? Very one-dimensional because it doesn't tell me enough about how different drugs interact under different permutations of drugs and doses. But nonetheless, if you take millions of data points and you run through algorithms, you can probably find that combo 3 is better than the other 2. This is a matter of using data to find better combinations. This is not an optimization exercise. All right, so let's make it interesting. Instead of 4 drugs, let's look at 12 drugs. All right, here's a challenge. I mentioned earlier that drugs and dose both matter. So if you take 12 drugs, 12 is a small number, and then you take 10 drugs, 10 is a small number, and you try each drug at 10 doses, that's a trillion possible drug combinations. Not resolvable no matter what resources you have. I've talked to people at Pfizer, Amgen, friends at Lilly, et cetera, Gilead, and they openly disclose they do not have the resources to do this type of work, especially when you're thinking about combinations. Let's make it even more interesting. Let's pretend that drug number 5 doesn't work on its own. Single drug assay, drug number 5 doesn't do anything. Traditionally, we don't think about drug number 5 again at all, right? But you know what? Let's leave it in. Because if we talk about optimization, even though a drug that doesn't work on its own is typically thrown out, it may in fact be a key component of combo number 1 out of a trillion. If you co-deliver it with another set of drugs at the right respective dose. And maybe at that point, it's not even about mechanism of action. It could be about unforeseeable synergies that exist that cannot be predicted, right? And so to get this information, we don't use pre-existing data. I want to emphasize the work that we do is small database, and we don't use millions of data points, we use hundreds of data points. But to get these data points, we have to run prospective experiments using multiple drug dose permutations. Right? And these are permutations that allow us to outline the parameter space, the galaxy of different options that exist. And you can't do that with fixed dose regimens. I need to know how drug 2 and 8 interact. 3 interacts with 6 with 10, 4 with 7 with 11. I need to design the experiments properly to cover every boundary of this space in order to properly interrogate the possibilities, except I don't need to run a trillion experiments. I only need a few hundred, all right? And when that happens, this is obviously schematic, but more often than not, we find that drugs that are not that useful on their own actually end up in the top ranked combination, but at completely unpredictable drug compositions as well as doses, all right? So I'll tell you about how we do this. All right, now you'll notice these plots, and this is work that I've done with many people, ranging, even collaborating with my father all the way to Ed Chow at NUS, who I've known for a long time. Now, and many others, the clinical community, as well as even, mathematics to clinical to biological. Now, if you look at what I've just told you, this is an in vitro study, what I just talked about. And people always ask, can you do this in vivo? The answer is yes. We can re-optimize the dosing parameters from in vitro here to in vivo. And we've done it in human as well, all right? So this is a process that has to be iteratively rerun at each stage. And when I tell people, I say, clinical trials don't fail in phase 1, phase 2, or phase 3. They fail here because 99.9% of the time, the combinations that are advanced into clinical development are suboptimal. Quite frankly, because if you take larger pools of drugs, it is impossible to predict purely using mechanism of action the best possible combination. But again, this is a process that we run from in vitro to in vivo to human. And something I want to emphasize is we've gone directly from in vitro and ex vivo straight into human before with astonishing outcomes. And people always ask us and say, hey, how can you go from here all the way to human? One thing I'll mention is that when we pick a combo from here, we are picking number 1 out of a trillion. We're not picking combo number 1 out of 1 or 1 out of 5 or 1 out of 20. We are finding the best possible combinations you can get from these insurmountably large parameter spaces. So we're already putting our best foot forward. That's a very different outcome. And not only that, these assays here don't just give you an optimal combo, they allow for positive and negative predictivity as well, right? Because again, this is more than AI, this is actually an optimization exercise. Now I mentioned we do hundreds of data points. How do we extract the optimums out? That's based on this curate correlation, all right? This is where we run this quadratic response to find global optimums from very small amounts of prospectively acquired data. All right, and this has been validated in 40 plus disease indications ranging from in vitro to in vivo to human. Here's examples of two patients or two different studies of patients that I'll talk about. One is HIV, 10 HIV patients dose reducing to find better administration protocols to reduce long-term side effects while maintaining undetectable viral load. And this is a liver transplant patient where we optimized immunosuppression dosing. All right, so I'm going to give you the use cases specifically here. About 5 years ago, we published a paper. This is clinical interventional prospective of using curate to dynamically modulate dosing. And you can see here, this is a single patient, 1 week, 2 week, 3 week, 4 week. Target trough levels you want to reach, different drug interactions over time. Not a huge surprise, by the way, for transplant that you have to titrate because it's commonplace to do that. However, this provides a lot more directed specificity and accuracy. And if you look at these three patients we sample, they look so different from each other over time. But in the randomized study, small cohort, we had the higher average MELD score, but we discharged our patients a month earlier compared to standard of care. For transplant, depending on where in the world you are, patients are often discharged 2 weeks after transplant. This was done at UCLA, and they treat the transplant patients that traditional transplant centers will turn away, right? These are the hardest to treat patients in the country. We discharged them a month earlier, which is good because you're removing patients from scenarios of community-acquired infection, and we outperform the control arm in every other metric, from time in target, time out of target, etc. Not long after that, we were contacted by a prostate cancer patient who was responding to treatment, PSA as his marker, coupled with imaging. 2-drug regimen. This is a BET-bromodomain inhibitor, which is in phase 2 trials, and an approved enzalutamide. Luckily, there was enough dose modification for us to solve for his quadratic surface. If you take a bird's eye view of this surface, to get the lowest PSA, we actually recommended a 50% dose reduction to the clinical team. Naturally, that was challenging for the clinical team, but we did it. The patient signed the waivers, and within 1 week, the PSA levels were the lowest they'd seen. This single data point was put into this plot. There was no change in the plot, so we kept the dose low again, and the PSA dropped even further. Remember now, for this one, we're not optimizing drug exposure for trough. We actually can optimize directly to efficacy as well. And this is further backed by the CT scans, durable response, and an active lifestyle for the patient. Next up, we're going to be advancing this to Neuro Oncology. As we know, for brain cancer patients, they have very few options once they fail temozolomide. With Totam Boone, Ed Chow, Bryce Tin, Andrea, TTO, Neuro Onc and Onc team, and neurosurgery team, we've already started running patient samples and very actionably, within days, finding better treatment regimens for them. I actually just got the trial protocol emailed to me about 20 minutes ago. We're about to embark on a prospective trial to evaluate this. This is a case study that we did, organoids established within 4 weeks, and then optimization within a week, finding better combinations that outrank traditional temozolomide. As you can see here, there's many options that outperform. Clinicians' choice for what to give, but we're excited to initiate this out of the Institute, and we'll see how that goes. All right, so to end off, I want to talk about COVID. So in November of 2019, right, so this is not last year, this is the year before, November of 2019, at N1, we convened a team not only of scientists and engineers, but of global health surveillance experts, Falvini and Raphael. They specialize in tracking outbreaks, for example. Gene Olinger, who developed some of the earlier Ebola combination cocktails, UC Kepo at NUS Business, Chris Hardesty at KPMG Singapore, to develop a plan in terms of, it's inevitable that we're going to get a pandemic someday. What use do we have for AI if that happens? And we published this paper around, I think it was April or May of 2020. We started this technical study in February. And within 2 days, we actually resulted in pretty cool answers on how to optimize combo therapy. But that paper also includes a lot of discussion about, and our conclusions are that economic development of treatments will be absolutely vital during pandemics because healthcare systems and industry will be overwhelmed, right? And so you need something that's quick and fast. And so that's where this work came from. And after that, a couple months later, we ended up applying this to the coronavirus. And I did a paper kind of highlighting some of this work as well. And what was interesting about this is, the team for that was interesting. We had people who worked at the CDC, and we brought them in to actually help us pick drugs that were globally available, right? When COVID first started, some of the earliest clinical trials with Kaletra and other drugs were very quickly established. Virtually no inclusion-exclusion criteria, outcomes that were all over the board, right? But at the same time, we picked a bunch of drugs, including Kaletra, right? Including, other drugs like azithromycin, Tamiflu and beyond, remdesivir, favipiravir, ribavirin. And we certainly ran EC50s, but we actually don't use the EC50 data to judge dosing, right? We actually use Cmax as a reference for dose ranging. Again, we run our optimization studies. And then from these results, we come up with ranked combinations, we study the interactions, we revalidate the findings, and then we produce an actionable list for doctors, all right? Our take-home findings from this were that remdesivir and Kaletra was the top-ranked combination. What's interesting to note is that Kaletra in our assay was relatively useless, all right, ineffective. And this mirrors with what the clinical trial results show. Yet, this is one of the earliest combinations that was widely used for COVID, right? But in our assay and in trials, we see that it's relatively ineffective. However, we don't use mechanism. We don't use even synergy to predict our optimization. We actually just arrive at the optimum first and then figure out why. And so when we went backwards, we found that lopinavir had an unexpected interaction with remdesivir. So just these two drugs alone dramatically boosts remdesivir's efficacy. In fact, remdesivir alone is not that good, all right? And so remdesivir is not that good. Kaletra is virtually not effective at all, but you put these two together and huge efficacy happens, all right? And the reason is lopinavir likes remdesivir. We know lopinavir likes ritonavir. So when you put the three of these together, we go all the way up to virtually 100%. And so the value here, and this is taking it all the way back to the beginning, after we find promising drug candidates, we still have to put them into combinations to maximize the utility of those repositioning candidates. And beyond that, the pity here is when early trials resulted in Kaletra not faring well, we largely stopped thinking about Kaletra. This happens every day in repositioning studies. Just because drugs don't work that well on their own, it does not mean that we should forget about them. But we will need to put them together with other drugs at right respective doses. We are forgetting about too many drugs that could be useful, right? But it's understandable. The parameter space is too big, right, to resolve unless we run an optimization exercise. So the final couple slides, we don't only yield one combo. We have a ranked list of combinations. We can query the list however we want. We can do top 4 drug combo, top 3 drug, top 2 drug, monotherapy. Remdesivir is okay. Kaletra, not really effective at all, but you put them together, you get virtually complete inhibition. That list is not just 30 some odd, 26 options. It's actually about 10,000 different combinations that were derived from the quadratic optimization. So this is available online on our N1 website. where people can come in and plug in different permutations, click the button, and you can see what the resulting efficacy as well as corresponding tox is. Again, running this study took 2 weeks. In 2 weeks, we had actionable combinations, right? And it doesn't only tell us which drugs are optimal, it tells us which drug combos should potentially be avoided as well, right? And if we had done this in January, many of the combinations out there probably could have been avoided, which matters when patients don't have months, but they only have days. All right, and so final thing I'll mention is where to. We have some pretty cool clinical protocols that have been approved. One involving a combo that we derived, and one to use Identify as kind of a first line clinical decision support platform. And we're going to start continuously running Identify now on different strains of the virus using different pools of drugs. And we're going to keep adding to that database to see if there's an evolution in how the combos are doing. We've since been backed by Sun Life and Micron Foundation to keep this work going. Final thing I'll say is that it's not all about the technology. We banded together as a team from both institutes, senior administrators, our communications offices, like 40 people. We came together to develop this book. We actually started this book 2 years ago, actually over 2 years ago. We knew that we've done AI for other diseases, but actually, no, we did nano for other diseases, AI for the first time, and we wanted to do infectious diseases. Available for free download at the N1 website, and it's also at all the libraries in Singapore as well. Final thing I want to say is ending on a personal note. I've been here now for about 2.5 years, and not long after we moved here, we had a family emergency. My wife was diagnosed with a brain tumor about 5 months after we moved to Singapore. And the community here has been amazing and effectively saved her life. And to give back to the community, we established what we call the N1 Wisdom Patient Impact Fund. And we seeded that with a personal contribution. And our donation is there to help fund MRIs, blood draws, clinic visits for patients here. And we did this because after, through the many clinical trials we've run, in our institute, we've learned a lot about the concerns of patients. If we kind of put aside the disease, put aside the medicine, put aside the technology, when we start to learn about patient concerns, we learn that patients are concerned about visiting another hospital because of the extra cost of a taxi ride. That they're scared to come in to get an extra blood draw that may give very valuable info on how to tune their dose, because of a taxi ride and the cost of a blood draw, or maybe at the cost of an extra image. And we don't want them to worry about that. So this fund has been populated to help cover the cost of patients here. So it's our way of giving back. And it actually took an entire collaborative effort to get this fund set up. And so we were very grateful to do that, and an exciting part of the institute as well. That being said, that concludes my talk. I wanted to thank all the people that make this work happen, all the funding agencies that support our work. And with that, thank you for the invitation to speak today.